My name is Tiana Jameson, and I'll be doing the cell cycle. Okay, so basically, life is based on the ability of cells to reproduce or make copies of themselves. This is done by a process called cell division, which is when one cell divides into two cells. For single-celled organisms, cell division is like reproduction. Each cell division not only produces a new cell, but it also produces a new organism. And an example of this would be bacteria or protists. For multicellular organisms like humans or us, cell division allows an organism to grow and develop from a single cell to trillions of cells, and also to repair and replace cells.
The cell cycle is a complex series of continuous events consisting of interphase and mitosis. During interphase, the nucleus and the darker staining nucleolus can be clearly seen. The DNA is copied. The chromosomes cannot yet be seen because they are still in the form of uncoiled chromatin. In animal cells, the centrioles duplicate themselves. In prophase, the chromatin coils to form visible chromosomes. The nuclear envelope and nucleolus disappear, and a spindle forms between the pairs of centrioles, which have moved to opposite ends of the cell. In metaphase, the chromosomes move to the equator of the spindle, with each chromatid attached to a separate spindle fiber by its centromere. During anaphase, the centromeres split, and the sister chromatids are pulled apart to opposite poles of the cell. Each chromatid is now a separate chromosome. Telophase is the final phase of mitosis. In telophase, two daughter cells are formed, with a complete set of chromosomes at each end of the cell. The cytoplasm divides, the nucleolus and nuclear envelope reappear, and the chromosomes begin to uncoil. When the new cells are separated, they enter interphase, and a new cycle begins.
Okay, here's a picture of mitosis, which is the fourth stage of cell division. Here, mitosis is divided into usually 4 steps, but we'll just go ahead and start with the first step. So here is prophase, and in prophase, the chromosomes are condensed, and then they attach to the spindle. And enter the prometaphase. In metaphase, chromosomes align at the center of the cell. Anaphase, chromosomes separate apart from each other. In the telophase, the chromosomes relax, the nucleus reforms, and the cell pinches into two and then splits by cytokinesis, usually ending in 2 daughter cells. So in conclusion, during the mitosis, the spindle fibers attached to the chromosomes pull sister chromatids apart, and then 2 daughter cells or daughter genomes are separated. Okay, so we know that mitosis is relatively short, and the cells don't usually spend a lot of time in the mitosis phase. A cell spends a good part of its life working and growing, breaking down sugars, making ATP, synthesizing proteins, enzymes, and other macromolecules. At other times, the cell makes preparations for reproduction or cell division, which is where one cell will divide to form 2 daughter cells, and the whole process will start all over again. Now here we see that majority of the time is spent in interphase, which is composed of three checkpoints, which is G1, S, and G2. And usually the overall length of the whole cell cycle is about 18 to 24 hours. We know this as generation time. Okay, so during G1 is known as the gap phase, or gap phase 1, is the primary point at which a cell must choose whether or not to divide. During this phase, the cells grow larger. The cells check whether or not the internal and external conditions are right for the cell to divide. They look at different factors like the size, is the cell large enough to divide, nutrients, does it have enough nutrients or energy available to divide. They look for the signals, molecular signals. Is the cell receiving positive cues such as growth factors? And they look at DNA integrity. Is any of the DNA damaged? Like, is it healthy? Now, factors can change depending on the type of cell. If the cell doesn't get its specific cues, it may cause the cell to enter the G0 phase, which is the resting or waiting phase. Now, some cells stay permanently in the G0 phase, while others resume dividing depending on if the conditions improve. So, like if the DNA is damaged or if something is wrong with the cell, it might just stay permanently in G0 and it won't do anything until it's the stuff that's in the cell gets better. The S phase, that's after the cell passes the G1 checkpoint and enters the S phase, it becomes irreversibly committed to division, which means it can do nothing but divide. This is known as synthesis. And during the synthesis, the genome is replicated. Now, G2 is known as gap phase 2. Now, gap phase 2 is the second growth in which the cell separates the newly replicated genome and marks the end of interphase, which is when it enters into mitosis. Here is Watson and Crick's model of DNA replication, and basically this whole diagram is an example of the semi-conservative replication, which is when half of the parent molecule is retained by each daughter molecule. And here you see that the strands unwind and each parent strand becomes a template for the synthesis of the complementary daughter strand, which coordinate together by base pairing here, which you see the G, the C, the T, and the A. They all stand for adenine, thymine, cytosine, and guanine, and these are called the nucleotides. Okay, here's a picture of eukaryotic replication. You see that the linear chromosomes are initiated at multiple sites, 1, 2, 3. And at the center of each replicon is the DNA sequence, or the origin of replication. And this is where the synthesis takes place and initiated by the initiator proteins. The origin recognition complex binds to the replication order. Okay, and this is a picture of bacterial replication in circular DNA. Here the replication forks are moving away from the origin. They unwind and the DNA then copies both strands. This is called theta replication. The two copies of the replicating chromosomes bind to the plasma membrane at the origins. When the replication process finishes, the cell divides by binary fission, and that's what this is. Okay, so some of the role of CDK, which is cyclin-dependent kinases. Um, they basically have many roles, but they're made early in the S phase or the synthesis phase, and they help activate DNA synthesis at already licensed origins, and they also prevent origins from being licensed again. Like they can't be made again after the CDKs are involved. DNA polymerase is an enzyme that can copy DNA molecules. Incoming nucleotides are added to the 3-hydroxyl end of the growing DNA chain. So elongation occurs in the 5' to 3' direction. Well, 5' to 3' direction. And several other forms of DNA polymerases have been identified, which are DNA polymerase 3, you see DNA polymerase alpha, primase, helicase, all that. So during the previous slides, we looked at the different checkpoints like G1, G2, S, mitosis, all that was involved in the regulation of the cell. And we know that the cell cycle is regulated to meet the needs of each cell type and the organism. But when cancer is involved, the cancer cells basically are a disease of mitosis. They rebel against the regulation of cell division. The normal checkpoints regulating mitosis are ignored and overridden by, different cancer cells. And the cancer begins when a single cell is transformed or converted from a normal cell to a cancer cell. A lot of times this happens because the cell, the function of the cells are changed, and one of several genes that normally function to control growth are messed up. Once these cell cycle genes start behaving abnormally, the cancer cells start to proliferate, and become repeated over and over again, uncontrollably. And unlike normal cells, cancer cells ignore the usual density-dependent inhibition of growth, and they multiply after contact with other cells, piling up nutrients, piling up everything that is involved with the cell, just basically becoming overflowed with nutrients until the cells are exhausted. And then it basically creates tumors, which are when the cancer cells proliferate to form mass, and as it grows larger, it begins to release protein from the cell to attract new blood vessel growth, which is called angiogenesis. The tumor types can range from benign or malignant. Benign is, of course, smaller. They remain at their original state. And malignant are when it gets serious. The cells send out signals that tell the body to produce new blood vessels at the tumor site. These cells not only have food and oxygen supply, they also have an avenue for escape to a new body part. And this is how tumors, can start, in your leg somewhere and move up to your heart. And they do this through the new blood vessels and into the bloodstream. And the cells that break away from the tumor begin to spread in the surrounding tissue. And, they do this by, bloodstream. Everything is transferred through the bloodstream. And when they start new tumors through the bloodstream, it's called metastasis.